Have you ever thought about extracting DNA from anything living? Welcome to your own channel that gives you power in the form of ADP for learning. Today we will see how we can extract DNA from living cells. First, you need to find something that contains DNA. Since DNA is the blueprint for life, everything living has DNA. For this experiment, we like to use green split peas. But there are lots of other DNA sources too, such as spinach, chicken liver, strawberries, broccoli. Specific sources of DNA should not be used, such as your family pet, Fido the dog, your little sister's big toe, bugs you caught in the yard. The extraction of DNA involves 4 steps. Step 1. Blender insanity. Put 1/2 cup of split peas, 1/8 teaspoon table salt, and 1 cup cold water in a blender and blend on high for 15 seconds. The blender separates the pea cells from each other, so you now have a really thin pea cell soup. Step 2. Soapy peas. Pour your thin pea cell soup through a strainer into another container. Then, add 2 tablespoons of liquid detergent and swirl to mix. Let the mixture sit for 5 to 10 minutes. Pour the mixture into test tubes or other small glass containers, each about 1/3 full. Now, the question arises, why we are adding detergent? The answer lies in the fact that blending can separate the pea cells, but each cell is surrounded by a sac or the cell membrane. DNA is found inside a second sac or the nucleus within each cell. To see the DNA, we have to break open these 2 sacs. We can do this with detergent. So, the question remains the same: why detergent? How does detergent work? Think about why you use soap to wash dishes or your hands. To remove grease and dirt, right? Soap molecules and grease molecules are made of 2 parts. Heads, which like water. Tails, which hate water. Both soap and grease molecules organize themselves in bubbles with their heads outside to face the water, and their tails inside to hide from the water. When soap comes close to grease, their similar structures cause them to combine, forming a greasy, soapy ball. A cell's membranes have 2 layers of lipid molecules with proteins going through them. When detergent comes close to the cell, it captures the lipids and proteins. So by looking at the figure you can easily visualize, after adding the detergent, what do you have in your pea soup? Step 3. Enzyme power. Add a pinch of enzymes to each test tube and stir gently. Be careful. If you stir too hard, you'll break up the DNA, making it harder to see. Use meat tenderizer for enzymes. If you can't find a tenderizer, try using pineapple juice or a contact lens cleaning solution. So the question arises, why are we adding meat tenderizers? The meat tenderizer acts as an enzyme to cut proteins just like a pair of scissors. The DNA in the nucleus of the cell is molded, folded, and protected by proteins. The meat tenderizer cuts the proteins away from the DNA. Step 4. Alcohol separation. Tilt your test tube and slowly pour rubbing alcohol that is 70 to 95% isopropyl or ethyl alcohol into the tube down the side so that it forms a layer on top of the pea mixture. Pour until you have about the same amount of alcohol in the tube as the pea mixture. Alcohol is less dense than water, so it floats on top. Look for clumps of white stringy stuff where the water and alcohol layers meet. So, at last, the question can come to mind: what is that stringy stuff? DNA is a long, stringy molecule. The salt that you added in step 1 helps it stick together. So what you see are clumps of tangled DNA molecules. DNA normally stays dissolved in water, but when salty DNA comes in contact with alcohol it becomes undissolved. This is called precipitation. The physical force of the DNA clumping together as it precipitates pulls more strands along with it as it rises into the alcohol. You can use a wooden stick or a straw to collect the DNA. If you want to save your DNA, you can transfer it to a small container filled with alcohol. Yippee! You have just completed DNA extraction. Now that you've successfully extracted DNA from one source, you're ready to experiment further. Try these ideas or some of your own, that is. Experiment with other DNA sources. Which source gives you the most DNA? How can you compare them? Or experiment with different soaps and detergents. Do powdered soaps work as well as liquid detergents? How about shampoo or body scrub? Or experiment with leaving out or changing steps. We've told you that you need each step, but is this true? Find out for yourself. Try leaving out a step or changing how much of each ingredient you use. Or do only living organisms contain DNA? Try extracting DNA from things that you think might not have DNA. Thank you for joining the fantastic journey of learning. In the future, we will meet with new innovative topics. Meanwhile, don't forget to like, share, and